Hello, my name is Bryn Me, and I will be going over the cytoskeleton and a couple of factors in it. And yeah, so first I want to go over the objectives. First I want to go over what they're made of, just kind of the basic of what makes up the cytoskeleton. I want to go over the function, and some of the three main ones, of course, are shape, integrity, and cellular movement. And then I want to look over some associated disorders or illnesses. I picked out two, but there are many more, just things that affect the cytoskeleton that, as science progresses, we're seeing more of a correlation with. And then I don't have it on this slide, but I want to go over what does the future hold. Okay, so first, there are three parts, three different types of categories of cytoskeleton. The first is microfilaments, aka actin filaments, which are about 8 nanometers or nanometers, or however you want to say it. And they are primarily made up of globular actin, protein polymers that often grow into a helical form that make what is called an F-actin chain. And all of these, of course, are going from smallest to biggest. And then there's intermediate filament, which is about 10 nanometers. That are mostly found, when they're found in animal tissue, they're normally as a cytoplasmic intermediate filament, but seen in some animals like fruit flies or insects, they have something called keratin intermediate filament, and intermediate filaments can be made up of keratin, neurofilaments, lamin, desmin, and yamentin, if I'm saying that correctly. And of course there's microtubules, which are about 25 nanometers, and they are cylindrical and hollow, and they're primarily made up of 13 proto-ligaments, and they go into alternating forms of beta and alpha tubulin, and you can see on the photo on the right here just how they are spaced off. You a lot of times will see microfilaments on the outside, and then intermediate there, and then microtubules are normally closer to the nucleus, as well as you can look down here, they enlarged it, that sequencing of the alpha and the beta tubulin. Okay, so the function of the microfilaments, first, they act as a track for myosin. The photo on the right really shows how that works, how myosin actually kind of walks on it and uses ATP to do that motion. And this allows for cellular transport and just allowing things to especially when they get into the cell, to have a starting area to walk to wherever they need to go. It also, because it is on the outside, adds pressure to the cell, and that can give it just some more structural stability, and as the next point, can help with cell shape. So if the cell is supposed to be a bit more flat or squamous or cuboidal, that can really help it, especially depending on if it's epithelial, if it's muscular, if it's skeletal. This allows it to stay in that form. It also can work in contractile forces in muscle cells, but also in non-muscle cells to help, movement, whether it be of the cell itself, or if it needs to work into a chain of moving a muscle group. This is kind of going down to what happens in the cell to cause that movement. And of course, I've already said cell transport, but then there's cytokinesis. in the telophase of mitosis and meiosis, microfilaments are used to split them, to create a barrier in the middle of the cloned cell and the now sister cell to have that distinction. The purpose of intermediate filaments, part of it is it's intermediate. It's in the middle of, if we look back 2 slides, it decided to zoom in, you could see it's right in the middle. The purple one, it's right in the middle of the micro filament and the microtubules, which allows partially for that structural stability to keep everything connected, but also just to have that interaction between those 2 different groups, and allows for that connection from the nucleus to the cell wall, or the cell membrane. It acts as a flexible intracellular tendon, practically. It allows for, if a cell needs to move, or it gets squished, or it gets stretched, it doesn't tear, it stays, kind of back into its own shape. It doesn't allow it to be easily destroyed. It keeps it from being stretched and damaged, and it also acts as an anchor for organelles. A lot of people think that organelles are free-floating in the cytoplasm, but that isn't true. There's a continual network of these different cytoskeletons that keep stuff anchored, not only to know where it is, but to make sure that molecules, proteins, and everything can get to it, rather than it just being free-floating and hoping that the molecules swim their way to the organelles. And there's the purpose of the microtubules. They are the biggest of them all, as we've seen. They also act as a track to transport stuff, especially between the nucleus and the organelles, and the organelles and the vesicles, and the nucleus and the vesicles. It really is like with the nucleus being the headquarters. Think of this as like an Amazon facility. These are the things, the roads that really help it go out from that head facility to wherever it needs to go. And if we're talking about something like an Amazon facility, you can see that is a typo, but the transport of kinesin and dynein, which are like the cars, except for they have to go in certain directions. Kinesin goes towards the positive end, while dynein goes more towards the negative end, as you can see in the image. So if it needs to go in one direction, it hops onto one of them. If it needs to go towards an organelle in the other direction, it goes there. If it needs to go back into the nucleus, it goes in that direction. It truly depends on where it needs to go. It also helps with cell shape and, of course, stability, especially with it being around the nucleus. These ones are very important for keeping the nucleus from being hurt. And then it helps with mitosis. In the stages of mitosis, when the chromosomes, or even in meiosis, when the chromatids have to have these spindles attached to them, these microtubules attached, they break apart the chromosomes from each other, or the chromatids from the sister chromatid or chromosome, which helps with, of course, cell division. And then, of course, kind of modified microtubules that have a little more differing to them are responsible for cell movement, like in flagellas and cilia. Flagellas being a bit more what we've seen in things like sperm cells, and cilia, where we see a lot of those in like intestinal tract cells, allow for increased space and movement of things across, or mucous membranes also have them, allow for small movement, and those are, of course, around the edges of cells, and then, of course, overall movement of the cell itself. You can see a lot of these also in the muscular and non-muscular cells. Okay. It was not moving to the next slide. There we go. To make sure that's the purpose, or the correct one. Okay, yep. Related illnesses. I chose, this first one is pretty close to my family. We've had people with this, so I chose Alzheimer's disease, which I didn't know had a correlation to faults in the cytoskeleton, but apparently it does. So when things like microtubules degrade, and they aren't being taken out of the cell, the material, the tubulins aren't being taken out, they can accumulate, and they accumulate as PHF, which is also paired helical filaments, and as you can see in the image to the right, in the tau neurofibrillary tangles up towards the closer to the nucleus of the cell, we see that buildup. Well, when that buildup happens, it damages the cell. It just doesn't allow the cell to work as properly, and it can lead to what we can see in Parkinson's is almost like a drying or shriveling up of the brain, and you can see those big caverns, especially in the center of the brain, kind of grow. And then I wanted to look into liver cirrhosis. I didn't realize that, the ethanol from alcohol. I specifically wanted to look into alcohol. I wanted to find one that isn't as much of humans' problems with Alzheimer's disease, but is more genetical. And I wanted to look at liver cirrhosis, which is a bit more, really hard to force someone or it's pretty hard to force someone to drink, so it's more about what the person does. But ethanol can lead to destruction of the cytoskeleton. It just kind of degrades at it. It can cause distribution of these cytoskeletons to change, whether it means it destroys them or it even gives them absences in different parts of the cell that are maybe closer to alcohol gateways or receptors. It has a harder time with expression changes, just how the cytoskeleton fully forms and what it goes to. And it has ability to do post-transitional modifications, which are never good to, after translation, really mess with that. It needs to go to where it needs to go. And then, of course, it reduces the removal of ethanol out of the cell, especially out of, in this case, hepatocytes, which then causes ethanol stress or alcohol stress on these liver cells. Okay. So what do we do with this knowledge? I say the great thing about science is it's always moving. Let's continue to do these scientific studies. Let's look at more into the cell. And you can see on the right here is a fluorescent dyeing or fluorescent view of the cytoskeleton, kind of showing what it looks like inside of a cell. I think we advance on that those studies. I think we do medical advances along with it, whether it be treatment. How can we treat people who have these disorders like liver cirrhosis and Alzheimer's and many different others, especially different forms of cancers that could arise from this? What's our next treatment for it based on that level? Detection, is there a way that we can, now that we know that a lot of these can be caused by cytoskeleton problems, do we look at signs for it, precursors, damage to the cytoskeleton that can kind of help us to treat before stuff happens? And of course, in the same area of prevention, what areas before we even need to detect them can people who might have a history of it or who have been drinking for a long time or all these different things, genetic predispositions, is there a way that we can give them treatment or find ways to help them before it even gets that far? And then, of course, all this comes down to increased understanding of the cell, what it's made of, what it does. We need to understand that the cell is not free-floating. It is very much connected with each other. That all of it works together in this kind of tension in a cell, rather than there being floating in cytoplasm. A review: the cytoskeleton is made of 3 parts: microfilaments, intermediate filaments, and microtubules. Their purposes: they can add structure, shape, movement, transport, mitosis, and even in meiosis. In cytokinesis, they are help with stability and anchoring of organelles and vesicles. There's a bunch of illnesses and disorders that can be linked to damage or malfunction of the cytoskeleton. It is a continuous network in the cell. Like I said, they're not free-floating. They're connected to the cytoskeleton, which makes it very important and allows for a higher efficiency than just things swimming around inside there. And kind of what we need to do next in science is we need to open up and look into doors that we didn't even know were there, and go further. And then helpful resources, I think, using LibreText or Khan Academy. This one I have LibreText. It goes over the cytoskeleton in good little chunks, and it has videos on how it works. It can always help people who are having a harder time or just need some more review of it. Look up fluorescent imaging, especially ones that have differing fluorescing colors of the different cytoskeletons, the 3 different cytoskeleton groups, just to kind of see how they interact with each other and where they are in the cell. And then I look up 3D models of the cytoskeleton. This link that I have actually is a shop for buying 3D models. But if you can find something like that and just look at them, and then maybe even going to find other 3D models to see how it works, because a lot of times we look into cells and they're very blank or empty because they want to show the organelles. But if you could find one that's a true, more realistic with the cytoskeleton, it will give you a better understanding of the pathways that the cytoskeleton has from the membrane to the nucleus to the mitochondria to other vesicles in the cell. And here are my links. Any of them are, I think, more than great to look at, to look further at different parts of the cytoskeleton. And thank you.